Assalamu alaikum everyone. So yesterday we discussed monosaccharides, right? That monosaccharides are monomers of bigger molecules like disaccharides and like polysaccharides, right? And we also discussed that there are three basic types of monomers that we will be discussing on monosaccharides. The ones that we'll be discussing are glucose, galactose, and fructose. Right? We also discussed that all three of them have the chemical formula C6H12O6. All of them have the same molecular formula. The difference arises in their structural formulas. And one by one, we discussed all the structural formulas as well. And we also discussed how they form or how they make the ring structures. And I told you that to stabilize themselves, what happens? These molecules, the open chains or the straight chains, they fold upon themselves, right? Hence giving rises to what? Ringed structures. So basically, we have ringed structures for glucose, galactose, and fructose, and that is how we'll draw them and represent them when we'll be studying them further, right? So the common glucose and galactose, although all three of them are hexose sugars, but because glucose and galactose, they have the double bond of oxygen attached to carbon number 1. Hence, they are all 6-cornered rings, right? Both of them are 6-cornered rings, whereas this one is a 5-cornered ring. So I'll draw all three of them one by one for you. Glucose and galactose are practically the same almost. So here, this is what glucose looks like, right? Every corner representing what? A carbon atom. We also discussed that if OH group is present below the plane, in 3 it will be the mirror image, right? And it will have here OH, H, and H, CH2OH, right? So if at carbon number 1, the hydroxyl group is present below the plane, we call it what? Alpha glucose, right? This molecule is called alpha glucose, right? If at the same position, carbon number 1, the OH group is present above the plane, rest is the same, right? Only if at carbon number 1, the OH is present above the plane, we call it what? Beta glucose. Now they're both glucose. They have the property of glucose, only the way they form bonds is different, right? They'll both have the same properties, like they're both water soluble, they're both monosaccharides, they're both sweet in taste. So because they're practically the same molecule with different arrangement, we use a term for them. They're both called isomers of each other. Alpha glucose and beta glucose are isomers of each other, just the way they form bonds in disaccharides and polysaccharides. That is different. Rest, they have all the same chemical properties, right? So moving on to the disaccharide molecule now. As the name indicates, di means 2, right? So when 2 sugar molecules, they join together by forming a bond, they form disaccharides, right? Like the name indicates, di means 2. Sugars, saccharon means sugar, right? So when 2 ring structures, they join together, they form a bond, they form a link, they form a disaccharide molecule, and then disaccharides lead to polysaccharides. So this is how this works. Monosaccharides, the basic unit, the monomer, right? Monosaccharides join together, they form bonds, and we'll see how, right? And they give rise to what? Disaccharides. Then disaccharides form further bonds and they give rise to what? Polysaccharides, right? So there are 3 disaccharides that you have to study in your syllabus, and all 3 of them, you must be familiar with them. There is maltose, there is lactose, and there is sucrose. We'll be studying all 3 of them. Maltose, lactose, and sucrose. Maltose is formed by the combination of 2 glucose molecules. You can say glucose plus glucose gives rise to what? Maltose. Two glucose molecules linked together, we'll see how. They form a bond and they give rise to what? Maltose. Glucose plus galactose, that gives rise to what? Lactose. So lactose is formed by two monosaccharides, glucose and galactose, right? Both glucose molecules linked together giving rise to maltose. Glucose and galactose linked together giving rise to what? Lactose. And glucose plus fructose gives rise to what? Sucrose. So this is how they're linked. So two monosaccharides joining together giving rise to what? A disaccharide molecule, right? Maltose, lactose and sucrose are all three of them are disaccharides. Glucose joins with another glucose molecule giving rise to what? Maltose. Glucose joins with galactose giving rise to what? Lactose. And glucose joins with fructose giving rise to what? Sucrose molecule, right? So I hope till here it's clear to all of you. Now I'll show you how glucose reacts with glucose. Now let's be very specific, right? Two alpha glucose molecules, because we've studied alpha glucose and beta glucose, right? So what? Two alpha glucose molecules, they join together giving rise to what? Maltose. So let me write the formulas or the ring structures for both the glucose molecules and then we'll see how they react, right? So here goes. Same goes for this one. Okay, so one alpha glucose, second alpha glucose, right? So alpha glucose here and an alpha glucose molecule here. So what happens now? You also need to remember the number of carbons, right? So if I start numbering, oxygen has number 1 here and number 5 here. So this is number 2, this is number 3, this is number 4, and this is carbon number 6, right? Similarly, oxygen has number 1 and number 5 on one side. So 1, 2, 3, 4, 5, and 6. So the reaction occurs between carbon number 1 and carbon number 4, right? What happens now? See, you have both OH groups or hydroxyl groups present here. What happens from any one molecule, an OH group, and from the other one, a hydrogen will be removed. A hydrogen and OH group will be removed, forming what? A water molecule. So a water molecule is removed, minus a water molecule, right? So when this water molecule will be removed, what will happen? This OH, and from here, this hydrogen is gone. Oxygen is left, right? That oxygen is going to form a bridge between the two molecules or the two ring structures, giving rise to what? A disaccharide. Two sugars now acting as one after the removal of the water molecule, right? So this is termed as the maltose. This is the structure of the maltose molecule, right? I'll write it down again for you and let's do it again together. OH here and OH here. What happens? A hydroxyl group reacts with a hydrogen atom, right? From one side, hydroxyl will be removed from any one side. So hydroxyl will be removed, and from one side what? A hydrogen atom will be removed. H2O, water molecule, is eliminated, right? And the remaining oxygen molecule forms a bridge between the two molecules, giving rise to a disaccharide or disaccharon termed as maltose. So this is how two alpha glucose molecules link together. Now they're no longer called alpha glucose. Now they're termed as what? A maltose molecule. This is the structure of a maltose molecule. And the reaction that occurs by the removal of water molecule, that reaction is termed as condensation reaction. What has taken place? Condensation. The reaction is termed as condensation. The removal of water molecule is called condensation. So condensation has taken place, right? Two molecules or two ring structures of monosaccharides, they join together, giving rise to what? A disaccharide termed as maltose. Okay, now this reaction is reversible. Just like by the removal of a water molecule, a disaccharide molecule is formed. Similarly, by the addition of a water molecule back into this molecule, what can be formed? They can both be converted back to alpha glucose molecules. So if I add water up into this, it was removal of water. If I add water, H2O, right? I can write it like this as well, HOH, this is water, right? So in one side, OH will be added. On the other side, also OH will be added, like H will be added, O will already be present, right? So by adding this molecule back, I can break this molecule up again into two alpha glucose molecules, right? This reaction of the addition of water, this is termed as, when I'm adding water, this is termed as hydrolysis. In the presence of water, the molecule is breaking down. Hydro means water, lysis means break down. So in the presence of water, the disaccharide is breaking down into its components, right? Into its monosaccharides, into its monomers. Therefore, the process is termed as hydrolysis, right? I'm repeating this up. What happens? By the removal of these molecules, OH and H, right? What is going to happen? Oxygen bridge is going to be formed, and the reaction is termed as condensation reaction. So by condensation reaction, two monosaccharides join together, giving rise to what? A disaccharide molecule. This reaction is reversible. By the removal of water, a molecule is linked together, a disaccharide is formed, and a disaccharide can be broken down into its components, monosaccharides, by the addition of water. So removal of water is termed as condensation, and addition of water is termed as hydrolysis. By adding water back, we can split it back into its monosaccharides, right? So this is how a disaccharide molecule is formed, right? And this bond has a name. This bond is termed as, the bond between sugar molecules is called this bond is called a glycosidic bond. The bond that is formed between two sugar molecules, linking them together, forming polysaccharides or disaccharides. That bond has a name, and the bond's name is a glycosidic bond. So a glycosidic bond is formed as a result of condensation reaction, and the glycosidic bond is broken as a result of a hydrolytic or a hydrolysis reaction, right? I hope this is clear to all of you. So I'll just give you a quick summary how maltose, lactose, and sucrose, all three of them are formed very quickly. I'm erasing this up. Okay. So what happens? Maltose you've already seen. Maltose is formed by glucose plus alpha glucose, right? Two alpha glucose, you add them up, what is formed? Maltose is formed. Now, lactose is formed by glucose plus galactose, right? So I'll draw this molecule. right? I'm not writing anywhere OH now, only on carbon number 1. And the lactose is the same practically. So this is carbon number 4, this is carbon number 1. OH from one side, H from the other side, and what is going to be formed? An oxygen bridge in between, right? The only difference was at here, carbon number 3 only had OH inward and H outward. This one had OH here and H here. Here, carbon number 3 and 4 both have the same property, right? So this is how the reaction takes place, right? Between alpha, like it practically looks just like maltose. The difference occurs where? In sucrose. Now, because in the case of sucrose, we have a fructose involved. In the case of sucrose, that is glucose plus fructose, right? Now, the glucose is alpha glucose, but the fructose is beta fructose. It is alpha, but this is beta fructose. Now, what is beta fructose? How fructose forms a ring structure? Let's just first look at that, right? So if you remember the structure of fructose, it looks something like this. The double bond is present here, right? So it had number 3 OH, sorry, number 3 had H here and OH here, OH, 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 OH. So this is the structure of fructose. I told you folding occurs where the double bond is present. Now double bond is not present at carbon number 1 now. Double bond is present at carbon number 2. So that means the link will be formed here and here. Carbon number 1 will be outside the ring and carbon number 6 will also be outside the ring. So the ringed structure looks something like this. There is this oxygen molecule, right? Linked with carbon number 2 and carbon number 5, right? Then there's obviously carbon number 3 and there's carbon number 4. So the structure looks something like this. It does not look like a glucose molecule. Where do carbon number 1 and carbon number 6 go? Carbon number 1 is also outside the ring and carbon number 6 is also outside the ring. Right? So carbon number 1 has CH2OH, carbon number 6 has CH2OH, right? Now, carbon number 2 had only one oxygen attached to it, no OH. Carbon number 3 had what? OH and H. Carbon number 4, H, OH. Carbon number 5, it had oxygen on one side and hydrogen on the other side. That's how this is present, right? Now, this is alpha fructose, the normal fructose that we have. But in the case of the sucrose molecule, we have a beta fructose. Beta fructose is this molecule, the same molecule, reversed only, upside down and at 180 degrees as well. So that means beta fructose will look something like this. Right? This oxygen comes upside down, right? Also, this was carbon number 1 and this was carbon number 2 over here. Now we have carbon number 2 on this side. That means carbon number 1 is present here, CH2OH, right? I hope it is clear to all of you, right? Okay, CH2OH and carbon number 2 also had OH on one side, right? So here, OH. Now, the rest is the same. Carbon number 2, carbon number 3, carbon number 4, carbon number 5, and carbon number 5 has carbon number 1 outside here, right? So I've done nothing. I've just shown you the structure of the beta fructose molecule that has been rotated upside down as well as at 180 degrees. This molecule, This is the alpha fructose, and this is what? The beta fructose, the isomer of this alpha fructose, right? So the reaction takes place between the OH of carbon number 2 and you have alpha glucose. So I'll draw an alpha glucose molecule here. Carbon number 1, OH and H, right? So here carbon number 2 has OH and here carbon number 1 has OH. The reaction is the same, the condensation reaction. OH from one side, H from the other side. The link to be formed between the carbon number 2, O, and the carbon number 1 on this side, this molecule is termed as the sucrose molecule. The reaction is between beta fructose and alpha glucose, right? I'm repeating the structure of beta fructose again. Why does it not look like a glucose molecule? Because it's a keto sugar. The double bond is present with carbon number 2 and the folding occurs or starts from the point where the double bond is present, right? Therefore, carbon number 1 and carbon number 6 are both outside the ring. The ring is a 5-cornered ring, whereas in the case of the glucose, the ring is a 6-cornered ring because folding starts from carbon number 1. So you have to add 1 atom up. Here we are removing 1 atom outside the ring, so the ring becomes 5-cornered. Because the ring is 5-cornered, see here the ring was 6-cornered. Still 1 carbon atom was outside the ring because 1 corner is occupied by the oxygen atom. Similarly here, the ring is 5-cornered, but both carbons are outside because 1 corner is occupied by the oxygen molecule that's linking both carbon number 2 and carbon number 5. So oxygen is shared between carbon number 2 and carbon number 5. Carbon number 2 drags the hydrogen out of carbon number 5. Hence carbon number 2 has an OH group and carbon number 1 attached to one side. Carbon number 3, carbon number 4 normal with OH and hydroxyl groups. Carbon number 5 on the other hand has no hydroxyl group because it has lost its hydrogen to carbon number 2. So it is only attached on one side with oxygen and on the other side with hydrogen and on the third side with the carbon number 6. So this is what this ring looks like. Here I have reversed this ring, converted the alpha fructose into what? Beta fructose. The ring has turned or tilted upside down. The oxygen has come upside down. So this structure is termed as what? Beta fructose. The reason we are studying beta fructose is because in the sucrose molecule, alpha fructose does not form the bond, beta fructose forms the bond with alpha glucose. So alpha glucose and beta sucrose or fructose, they link together forming the glycosidic bond in between with carbon number 2 and carbon number 1 involved in the reaction. Condensation takes place and what is formed? A sucrose molecule is formed. Sucrose is the common table sugar that you use in your everyday life, right? The sugar that we eat, that's actually sucrose, right? So this is all about it. All the disaccharides, they're all sugars, they're all sweet in taste, they're all water soluble. There's only a slight difference, one more topic that we'll be discussing today before we begin with polysaccharides, and that is reducing and non-reducing sugars. I think you might have studied that before already in O3, but we'll be doing it in a little bit detail. So reducing sugars and non-reducing sugars. So as the name indicates, when these sugars react with certain substances, they reduce them, right? The same reduction that you studied in chemistry. So when these sugars react with certain chemicals like copper sulfate, they reduce them and in the process, they are themselves oxidized. All the reducing agents that you might have studied about, when they're reducing one compound, they are in the process themselves being oxidized, right? So the sugars that reduce, that are capable of reducing other compounds, those sugars are called reducing sugars. All the sugars that you have studied are reducing sugars. The glucose, the galactose, the fructose, maltose, and lactose. These are all reducing sugars. There's only one non-reducing sugar, and that is sucrose. Sucrose is the non-reducing sugar. Now, as you already know, what is basically reduction? Addition of hydrogen, addition of electrons, and removal of oxygen. That's reduction. So if any one of these three is occurring, we say that reduction is taking place, right? So to test the reducing sugars, we make them react with the copper sulfate solution, right? Which is termed as the Benedicts solution. The active agent in the Benedicts solution or the Benedicts test is done to check the presence, right? Now this is a part of your practical as well. So Benedicts test is done to check the presence of reducing sugars, right? If you have a mixture of elements or compounds, you have a mixture of proteins and lipids and sugars, and you have to confirm the presence of sugars, you'll first have to make sure whether it's a reducing sugar or it's a non-reducing sugar, right? So to make sure, what test do we conduct? We conduct the Benedict's test. How is this test done? We take the sample, right? And we take the Benedict reagent in equal quantity. Sample and Benedict reagent, they're mixed together, right? Let's say we're taking 2 ml of sample and we're adding 2 ml of the solution to it. And then we start heating it in the presence of water bath. Why? Direct heating can make it catch fire, right? So we heat it on a water bath, and then the color changes. The color changes from the Benedict solution is blue in color. The color changes from blue to green, green to yellow, yellow to red, sorry, yellow to orange, and orange to red. This is the sequence of the color change. The color changes from blue, which is the color of the Benedict solution, to green. Slowly, very slowly this occurs. From green to yellow, yellow to orange, and from orange to red. Red is the final result. Even orange is considered the final result. So if this color change occurs, that means reducing sugars are present. Now you need to know the chemistry behind it. What is actually happening? In the case of the Benedict's test, the Benedict's solution that is blue in color, that contains copper sulfate. Copper sulfate makes it blue in color. Now, copper sulfate has the copper ions with the valency +2. When reducing sugar reacts with this copper, what it does, it reduces it to what? Copper 1 ions. So what happens? The reducing sugar plus copper ions, Cu +2, it is going to convert it to what? Cu +1. This is copper sulfate is blue in color. It is converting it to copper oxide, which is red in color. So that is why the color change occurs. Here, the copper is present in the form of copper sulfate. Here, it is present in the form of copper oxide, right? And the sugar itself is oxidized. So it is adding electrons here, therefore it is going to lose electrons on its own. So reduction of electrons is what? Oxidation. Hence, in the process, the sugar is itself being oxidized, right? So this is the entire chemistry of the Benedict's test. What happens when you add the reducing sugar to Benedict's solution? The active agent in Benedict's solution is copper sulfate, that is blue in color. When you heat it, you're speeding up the reaction, right? So what happens while heating, the color starts changing. The reducing sugar starts acting on copper sulfate, changing it to green, yellow, orange, and eventually red color, right? What actually it is doing is, it is reducing the copper molecule, copper +2 to copper +1. It is reducing the oxidation state or the valency by adding electrons. And in the process, the sugar is itself being oxidized, right? And I told you how we perform the test already. We take sample and we take the Benedict solution in equal quantity. We mix them and then we start heating it over a water bath, right? If at the end of the heating process, that is mostly 2 minutes, what happens? The color changes to red. we'll show that a reducing sugar is present. It can be glucose, it can be galactose, it can be fructose, it can be maltose, it can be lactose. What it cannot be is sucrose, right? So this is a confirmatory test. We also say that Benedict's test is not just a quantitative test. It can also tell you the quantity of sugar present. If the sugar is in less quantity, the color won't go further from yellow. If the color change stops at yellow, that means the reducing sugar were in lesser quantity than the Benedict's reagent, right? And if the color goes all the way to red, that means that the sugars were in a greater quantity present, right? What about also while doing experiments, we can also compare time. If this color change appears quickly, that means that sample has more reducing sugars. Please write it down somewhere, right? Two ways of checking the quantity of the reducing sugar. Number one is the color. You can compare the color, right? The darker the red color or the further the color change goes, the more is the quantity of the reducing sugars. Also, the more time the sugar takes to convert to this color, that means lesser is the quantity of sugars. जितनी ज़्यादा sugar होगी, उतनी speed से the color change will occur, right? तो ये टेस्ट तो हमने कर लिया. If you have present glucose, galactose, fructose, maltose, lactose, the reducing sugars. What if in the solution sucrose is present? What test you will perform for sucrose? Now, sucrose, as you know, is made up of two reducing sugars. Sucrose on its own is a non-reducing sugar, but sucrose is made up of what? Reducing sugars, right? So, to perform the test for sucrose, what we will do is, we will have to break the sucrose molecule down to its components. So to break it down, we'll do hydrolysis in the presence of acid. So we will add HCl to the sample solution, right? Basically what the HCl will do, it is going to break down sucrose to glucose and fructose, right? So glucose and fructose are both reducing sugars. So पहले आपने टेस्ट परफॉर्म किया, Benedict's test वो नेगेटिव आया. That means कि reducing sugar present नहीं थी. अब हमने we have to make sure कि उसमें sucrose भी present नहीं है. So for that, what we will do is we'll perform the test for sucrose. To do that, what we'll do is we'll take the sample solution, we'll add HCl, and that process will do what? Hydrolysis of the sucrose molecule. HCl is just speeding that up. So sucrose is breaking down into glucose and fructose. Now glucose and fructose are both reducing sugars. Now on this sample, we'll perform the Benedict's test, but before that, because it has become acidic, Benedict's solution does not change color in the acidic medium. So we'll have to neutralize it. To neutralize it, we will add a base that can be sodium hydroxide to the solution. And then perform Benedict's test. That is, add Benedict solution, heat it over a water bath. If now the color change occurs, that means कि sucrose present था, because sucrose has been broken down और अब वो color change दे रहा है, क्योंकि पहले उसमें कोई sugar नहीं थी, color change negative आ रहा था आपके पास, अब color change positive आ रहा है, that means कि आपने HCl डाल के क्या किया, sucrose को break down कर लिया. अगर इस दफा भी result के बाद no color change occurs, that means the solution contained no sugar and you can start testing it for the presence of proteins or for the presence of lipids, right? That's all for today. What I want you to do is please read it out. I'll mention the page numbers. From page number 30 to page number 32. You have to finish reading on your book from page number 30 to page number 32. On page number 32, you have a purple box that explains both of these reactions. You have to memorize them. You have to learn the chemistry behind them for your practical paper. One of the most common experiments that you get in your practical paper is the test for the sugars, right? Or somehow, even if you don't get it, you may get questions out of it. So please do go through that purple box, and if you have any questions, you can ask them tomorrow, right? Okay, that's all for today. Thank you.